Hi, everyone, and welcome to the first Sentinel Collective of the season. We're incredibly excited to have you all here, and we're really excited to bring a couple of guest speakers to you who are involved with some collaborations that you'll get to meet in a few minutes. My name is Rachel. I am the communications coordinator and a member of the Sentinel program team with the Be Informed Partnership. And I will pass it off to the other members of the VIP team to introduce themselves.
Hey, I'm Eric Malcolm. I work with the outreach coordinator. Work really closely with Rachel here and Michaela and Natalie for the Sentinel Apiary Program, and looking forward to working with you all this year.
Hi, I'm Michaela. I work on the database, and at the end of this presentation, we're going to look at the database again. Some of you may be familiar with it, and we can get you all set up. And if you have questions, when you send those in, I'm looking at whatever your questions are and help you all get set up. So thank you. I'll pass it on to...
Hi. I am Natalie. I am the science coordinator for the Be Informed Partnership. And yes, as Rachel and Eric and Michaela says, we're really excited to have you back for this new year of monitoring bee health. So this program is all about trying to make it easier for you and giving you kind of this accountability schedule and framework to help you monitor your colonies and provide also a network to, encourage some research on honeybee health data. So thank you for coming back.
Awesome. Thanks, everyone. So as I mentioned, we have two guest speakers here with us today. If you both want to introduce your like a really quick introduction of your work before we get into the presentations. Garett, you're first on my screen.
Sure. So my name is Garett Slater. I'm currently a postdoc at the USDA ARS Bee Lab in Baton Rouge. I recently got my PhD at Purdue University just about a year ago. I've been there, pretty recently. And so my research really focuses on honeybee genetics, specifically how can we use some of these modern genomic tools to improve honeybee breeding.
Awesome. Thank you so much. Zac.
Hi, everyone. My name is Zac Lamas. I'm a postdoc here in Jay Evans' lab at the USDA, but in Beltsville. And I largely study Varroa behavior. And when we identify a new behavior, we then ask, how does that behavior drive viral transmission? And that's the bulk of what encompasses my work.
Great. Thank you so much. Okay, so I'm going to go over some really quick, some housekeeping stuff before we get into their presentations. So first, I want to mention that as with last year, we are doing the NOCEMA in May and July only. Now this year, it's going to be more strict. So the only samples that are going to be processed for NOCEMA are those that were sampled in the months of May and July. So please be sure to get your samples in to make sure that you get all of your sample processing. All right, so we also wanted to announce one of the changes to this year is that we are now offering Sentinel participants add-on sample processing. So basically, if you have any additional sample analysis that you'd like to get this year, for example, additional Varroa or NOCEMA, or if you want to order separate pesticide or viral analysis, you'll be able to do so. The link to do that will be available soon once it's complete. And those order forms will be, or those order periods will be available in only the month of May and the month of July. And anything that you order will be shipped separately from your Sentinel kit, of course, because those have already been sent. And those samples will have to be taken and sent back to the lab in the month that they were ordered in. So that's really exciting. That's the first time this has been offered. And so we'll send out emails with reminders about when those order periods are available and when they're closing. And then Eric has one thing to mention, and then we can move on to the presentations.
Yes, and thanks, Rachel. So just very quickly, this year we're actually going to be doing a abbreviated pilot version of what the Sentinel Apiary Program is going to eventually, hopefully, morph into in future years to just help us continue to expand what we're doing with the community. So we've got a couple of clubs and organizations who have signed up for participating in this pilot. We're looking for 1 to 2 more people with either a teaching apiary or a mentoring yard that they use for a number of club mentees. So if that sounds like you and you would like to help be one of our kind of first cohort in this program, please reach out to us. Stick around after the meeting, and we'd love to talk with you a little bit about that. Thanks so much.
Great, thanks, Eric. All right, Garrett, again, you're first on my screen. You want to take it away?
Sounds good. So let me share my screen quick. Okay, you might have to let me share my screen.
Good to go.
Perfect, thank you. Okay, perfect. Well, so first, I introduced myself as Garett Slater, and I'm based out of the Bee Lab in Baton Rouge, and one of our main focuses is breeding and genetics, right? So how can we identify the best the genetics to provide the healthiest, most productive colonies? And we've been doing this for 56 years with the pole line and some of the VSH traits. And so one thing we wanted to sort of expand on is how can we go from some of this phenotypic selection, right, this traditional selection, to using genetics. And so I'm excited to kind of share my research and how, the Sentinel Apiary Program can really help with me and also hopefully help with you. So let's see. So first, when we think about what contributes to colony losses, it's sort of multifaceted, right? There's a lot of these different reasons and challenges that actually cause colony losses. And I kind of outlined probably the 6 major that are reported by the Bee Informed Partnership and other monitoring agencies, right? We have viruses. And so we know that there's certain viruses such as deformed wing virus, paralysis virus that can drive colony losses. Varroa is definitely number 1. We know that if colonies get above a certain threshold, those colonies are more likely to collapse. We know there's a wide range of pathogens associated with colony loss and colony health, such as European foul brood, American foul brood, and Nosema. And then, queen fertility. Queens failure is a major issue among beekeepers. There's been reports that about 60% of queens are failing within 6 months, where they should be thriving and reproducing for 2 to 4 years. And then there's land use changes with nutrition forage and agrochemicals. But we can usually split these into when we think about these challenges, what can we do as researchers in the beekeeping community to really address these challenges? And one thing that we can do is, genetic improvement. And so in the black, viruses, varroa, pathogens, and fertility, right? We know that there's a genetic component that can confer resistance or tolerance to viruses, varroa, and pathogens, and genetics that can improve queen fertility and drone fertility overall. And so one thing I've really been focusing on is, okay, we have these traits, and and you as a beekeeper, you've noticed these traits where you have 2 colonies next to each other. You notice that one may have low varroa numbers and one next to it may have high varroa numbers. And that could be partially environmental, but there could be genetic differences between those colonies. And so one thing I'm really interested in is, okay, we have some of these known tolerance traits across the United States. How can we identify the genes associated with these traits and use these to, overall improve colony health across colonies? And so one thing I'm really focused on is, what are the genetics associated with some of these traits, and how can we use this to improve colony health and productivity? And so why is it important to understand genetics? And so it really depends on your goal as a beekeeper and your background. And so for me, I can really outline these 2 major goals. So for breeders, what we want to do is use genetics to identify queens which produce the healthiest and most productive colonies. And so, if you're a queen producer or a queen breeder, this is such a difficult task because, you have hundreds of thousands of colonies. How do you actually identify the best queens that not only fit you as a breeding program, but also fit your clientele? Because, you're selling thousands of queens per year. How can we help breeders identify the best queens that sort of fit these challenges? But also, how can we help beekeepers purchase the best queens that have optimal genetics based on whatever your goals is? And so one thing we want to do is, how can we use genetics to really identify the best queens and help these 2 major demographics of beekeepers? And so first, I want to give a little kind of background of genetics, because I think it'd be an abstract concept, to a lot of beekeepers. And I gave a lot of talks about, just basic genetics of honeybee colonies. And so when we think about genetics, we think about, we have right, we have the DNA, the deoxyribonucleic acid, and these DNA is made up of these genes and genetic variants. And so here's an example of gene A, and you see there's these different nucleotides, G, T, C, and A. And basically, I mean, it's kind of amazing. The DNA in every single honeybee cell is about 6 feet long, but it's kind of stored in this compact form. And based on how this DNA is read, it can be expressed into these different traits. And so this DNA becomes a protein structure, and this protein structure becomes a trait, and this trait can be important for whatever trait you're interested in beekeepers. And so, for example, we know that certain genes can express these proteins that are associated with hygienic behavior. And so if colonies have higher expression of these proteins, they can have higher hygienic behavior than other colonies. And so one thing we really want to do is how can we connect the genetic level, right? We have these genetic variants to the actual trait level, whether it's hygienic behavior, honey production, or some of these grower defense behaviors. And so here's an example. And so when you think about DNA, there's these different markers, and these are named a lot of different things. We call them SNPs, SNVs, we call them markers. There are a lot of different names, but they're basically the same. And we can refer them to basically these individual nucleotides. And if these individual nucleotides have a mutation where they change from maybe a gene C to an A to a T, that can impact a trait. And so, for example, let's say this marker A, G, and C, this marker could be associated with high hygienic behavior. And if you're not familiar with that, that's basically this colony level behavior where you freeze 100 brood cells, and you come back the next day and see how many of those dead brood were removed by the bees. And so these markers can be associated with this trait. And so some research out of Canada show that there's about 186 of these that are strongly correlated to some of these traits. And so this is one way we can think about genetics and how this impacts some traits of interest. And so for you, some short term is, where do genetics actually come from? And so, you're beekeepers, and I'm sure most of you experienced, honeybees are non-native to the United States, and they come from, Europe and Africa and Asia. And so most of the genetic variation actually originates from, across the sea. And so when you think about the genetic variation and some of these mutations that could affect a trait, they're actually not arising in the United States. They've arose, thousands of years ago in Africa, Europe, and Asia. And so this is really where genetics actually come from, and what's really important for how this actually connects to the trait of interest. And so one thing we want to do in the short term, and I think this would be interesting to a lot of beekeepers, is what is sort of the genetic ancestry of your colonies, right? So we can sequence your entire colony, see, okay, what percentage of the genes come from Africa, Northern Europe, Western Europe, or Asia. And so here's an example of a genetic report that we plan on doing with the Sentinel Apiary Program, where we'll sequence a single colony, and we'll tell you what percentage of the genetics originated from these different regions. So, for example, for this example, this colony, 12% originated from Africa, and C and M are these European lineages, and so you can see a high proportion of that, and you also see some of these other lineages. And so one thing we want to do is tell you what your genetic ancestry to your colonies is, but also add in sort of mixture analysis, which shows you, okay, how much of your colony is basically a mutt. So what's the actual total mixture of it, and which one is it more pure? And we can tell that through this admixture score. And so 0.45 means if it's more of a mutt. If it's closer to 1, that would mean it's kind of a more pure. The next thing is genetic diversity. And so I get a lot of questions about genetic diversity and it being low in the United States. And so one thing we can do is look at the genetic diversity of your honeybee colony. And so that's going to be sort of a short term for you as a beekeeper, and so one thing to expect. And so long term, I really focused in the beginning of the talk, what is this concept of genomic breeding? And so how can we, use genetics to breed for more productive and resilient colonies? And so before I kind of give a background of these, different steps in breeding. And so every single livestock industry goes through these different steps. And, we call them breeding step 1.0, 2.0, and 3.0. And so right now, the honeybee industry is kind of in this kind of breeding 1.0. It's sort of more incidental breeding, where I wouldn't say there isn't any rhyme or reason to the breeding, but beekeepers aren't necessarily using statistics or using data to select their colonies of interest. It's kind of like, this queen seems more productive than this queen. So it's really more subjective. And so this is really what most beekeepers are using. Breeding 2.0 is when we start integrating statistics and more experimental designs to better choose the queens. And breeding 3.0 is when we start using genetics to breed from traits. And so instead of actually looking at the trait of interest, we can basically sequence a colony and predict whether this colony is more likely to produce more honey or to have more hygienic behavior expression or some of these other traits of interest. And so while most of these other industries, such as the dairy industry and these other livestock industries, are in this breeding 3.0, we're still in this breeding 1.0. And so we're still behind. And so one thing I really want to do is kind of work with beekeepers across the country and find, okay, what genetics are important for these traits and how can we use this in sort of a genomic breeding program? And so one thing we eventually want to do at the USDA ARS in Baton Rouge is to use its genomic breeding to make sort of this kind of rapid progress. And just kind of illustrate why we want to use genomics over traditional breeding. It's not like traditional breeding is bad and it doesn't confer these traits of interest. And so, I mean, for example, the USDA, we bred for, Varroa resistant traits, right? I don't think a lot of beekeepers know that. So we've done this in traditional breeding. But the one benefit of using genomics over traditional breeding is we can not only select earlier in the season, but we can select more often. And so, for example, for traditional breeding, there is this time and cost input to, measure the trait of interest. And so in order to actually measure most traits, you have to invest time and energy for your colonies to get to their maximum colony size. Whereas genomic selection, we basically sequence a queen or a small nucleus colony and predict whether the colony is more likely to have this trait or not. And that allows us to not only select earlier, but select 3 to 4 times in one season versus just one. And so it's not like traditional breeding is a bad thing. It's just genomics can really accelerate some of these trait selections, especially. This will especially be important as we kind of face some of these new challenges, such as new viruses or even the trophy mite. And so one thing we want to do for the long term is report breeding values. And so breeding values is just a really fancy way of saying, is your colony more or less likely to have this trait of interest? And so one thing we want to do is not only have this ancestry report, such as a genetic background, which you receive immediately, but also have a report about how likely your colony is to have this trait. And so, for example, is this queen that you're purchasing more or less likely to have Varroa defense behaviors, or is your colony more likely to produce more honey or have lower pathogen levels? And so this is one thing we want to do. And eventually we'll have a sort of a queen potential score where, an average is 0. So this would be the average across everything. A queen potential score would mean a plus 10. And so let's say you're sequencing, 50 queens and, there's 2 queens that have a plus 50 and other queens have a minus 50. That allows a beekeeper, whether you're a breeder or sort of a beekeeper, to choose the best queens and continue to select for that. And so that's one thing we want to do sort of in the long term. And with this genetic report, right? I mean, immediately we'll be providing this kind of genetic ancestry report, which I think is going to be really interesting. But as we collect more data, we can still spit more reports in the future. It's kind of like a 23andMe test in that way, where you genetic test once, but you always kind of continually receive more data as the research team and 23andMe team gathers it. And so what do we need from you? And I think this was sent out, or it's going to be sent out, but one thing we want to do is have you collect, 10 to 50 drone larvae. And one reason we want drone larvae is because they're haploid. They have, one set of chromosomes versus 2, which is diploid for females. We can use that to basically predict the queen genotype. I mean, if you want to send me your queens, that would work too, but I think drone make more sense for you. And so collecting drone larvae, whenever your colonies produce drones, I think it's to be kind of dependent on your region, but hopefully, May or June would be a really good time. And so in the kit, there should be a vial of alcohol. And so just putting drone larvae into the alcohol, and they have to be larvae. They could be pupa also, so that would work. 2 is just kind of monitor your colonies and collect the trait data. And so I know as part of Sentinel Apiary Program, you're required or you're supposed to collect Varroa alcohol washes and no semen, all that stuff. So monitor your colonies. Next is, I would like to give kind of updates on my research progress and things you want to do. And so eventually, long term, I want to sort of produce this, breeding network. And so at the USDA, we're breeding for all these traits. And so how can we produce kind of these small breeding networks across the United States to, better disseminate some of these stocks and better help beekeepers select some of these traits? And then finally, we'll give you a report. And so, like I mentioned, the short term is going to be sort of a genetic ancestry report. So just telling you about the genetic background of your colonies. But in the long term, hopefully we can tell you whether sort of a queen potential score and also whether your colonies more likely express some of these traits that we're looking at. And so, yeah, first of all, I want to thank the funding partner with North Dakota Department of Agriculture because they're funding really this whole project. And, I only had 10 minutes. And so I try to get through everything pretty quickly. And so if you have any more questions, comments, or talk requests where I can give you kind of a more extended talk about genetics and my planner, just feel free to email me. Yeah, and with that, I'll take any questions.
Awesome. Thank you, Garrett. I just want to jump in and say, yes, the tubes are in the kits, and I believe it is 1 tube per colony. Is that correct?
Yes. And so, yeah, I think we agreed on, so it's you could have 8 colonies per program, right? Or per
Either 4 or 8. So depending on what size kit you ordered, you'll either have 4 or 8 tubes in your kit. So you would just collect 10 to 15 per colony. Make sure that you label it, which colony it's from.
Yep. Yep. So you have the option to just do 1 colony, but if you want to do all 4 or 8, I mean, we're happy to sequence more. So
awesome. Thank you so much. And those tubes can just be sent back to the lab with your regular Varroa samples for the Varroa processing. All right. Questions for Garrett? And if you think of any questions later, you can always send them to me in the chat, and we can ask them at the end of the meeting.
This is Maureen. I just have one question. So is your preference for larva or is your preference for pupa?
it doesn't really matter. It just as long as they're in the developmental stage. And one reason we don't want adult drones is because there's so much drift between colonies,
Sure.
but we can't really tell exactly where they came from.
Okay.
But they're pupa or larva, that definitely will work. And the more the merrier. And so we can get 10 to 15, that would be great.
Okay. Thanks.
Yep.
It's a great question, Maureen.
I have a question.
Sure.
Can you collect the data from specimens that are in alcohol?
What data? Do you mean the genetic data?
Yeah.
Yes. And so we can preserve the DNA and the alcohol. Yep. And so the vials will be full of I think 100% alcohol, and so that should preserve the DNA long enough to get into kind of a cold freezer. And so yep.
Oh, okay. I was talking about historical data. So I have a really interesting set of data because I've never purchased bees or queens. All my bees originate from feral colonies.
Oh, interesting.
And I've been breeding from them for like 6 years using phenotypic selection based on Varroa levels. But I have a whole historical collection of anytime a queen dies or anything, I save her in alcohol, and I have all their associated mite data.
Oh, that's awesome. Yeah, I'd like to talk to you a little bit more about that because do you have a pedigree also? I suppose
well, I know my queen lines, but I'm open mate, so I don't know.
No, that's fine. As long as you know the queen lines, okay.
Yeah, no, I do have them.
Yeah, let's chat a little bit. But with the historical data, I mean, DNA is really stable, and so if it's in 100% alcohol, right, and so it should be pretty stable.
It's actually, I think it's a 90% rubbing alcohol.
Okay, yeah, that should be good. So,
and I also have a huge selection in just a general freezer.
And is it workers?
I have workers in the freezer, but I have the queens in alcohol.
Oh, you have the queens. Oh, that seemed perfect. Okay.
Yeah, no, I've actually, we definitely need to talk. I've actually talked to Jay about some of my bees in the freezer before.
Perfect. Yeah. Yeah, I guess you have my email, right? You can just contact me, and I'd like to talk to you more about that.
Cool.
Garrett, could you put your email in the chat?
Sure.
Awesome. Thank you. All right. Are there any other questions for Garrett? Nope. All right. Well, thank you so much, Garrett. And Zac, and just to preface, Zac's project, more information will be coming out about that in terms of how to relate to you. There are no sampling materials in the kits for that, but talk about that in a minute.
All right, Garrett, you have no idea how jealous I am that you work with DNA. Just hearing that you can, ship and take historical samples and alcohol. For everyone else, I work with RNA, so it degrades very quickly, and we can't get away with that. But anyways, let me grab my presentation. Okay, does everyone see the correct screen?
Looks
Perfect.
good.
Fantastic. Sorry about that delay. So I study Varroa behavior, and I'm going to quickly go through a presentation that normally takes a lot longer. I've taken a couple things out. But this slide is a spoiler alert. So for almost 30 years, we got a really prime piece of information about Varroa destructor incorrect, and we're now addressing this big gap in knowledge. We've said that mites prefer nurse bees over all other adult bees in the colony, and that turns out not to be completely true. Mites actually highly prefer an aggregate on young adult drones. This gap in knowledge partially arose because all previous studies that looked at host preference by Varroa destructor, and this is adult bee host preference, actually omitted adult drones from the experimental designs. All of these experiments were done in the laboratory. The only one that was done in the field was done in these very small colonies. And again, they only used worker bees. So they're studying host preference by different aged of worker cohorts, a newly emerged nurse bee forager, but they just simply didn't have drones in the experiments. I noticed this gap in knowledge, set up a quick experiment during the start of the pandemic, and saw that mites actually highly aggregate on these young adult drones. So we set up these field trials where we would mark newly emerged workers and newly emerged drones every day for 5 days and remark them and give them the very specific color pattern so we could track each individual bee if they acquired a mite or were born with a mite. And over on the left is one of my interns who, rain or shine, we would do these experiments continuously, marking for 5 days and monitoring for a total of 9 days. And this is what our data looks like after 2 years. Over on the left is our early season data, and over on the right is our late season data. Green is drones, orange is workers. My x-axis is the age of the bee, and my y-axis is the number of mites found on those individual bees. Now, most bees don't have mites on them. This is what you'll find in your colonies. Most bees are mite-free at the moment that you look at them. But when we do find detections, most of our detections are on these younger 1, 2, 3, and 4-day-old bees. Now, the 1-day-old bees, they emerged with the mite on them. We're collecting these at emergence. So this really represents the invasion rate into your brood. And we already know that mites prefer drone brood over worker brood, so that's not surprising. What was really surprising is drones that emerged without mites on them on day 1 would acquire upwards of 6 mites by the 2nd day. And this is because mites are actually actively moving from one bee to another within your colony. They're most attracted to these 2-day-old bees. Now, when drones disappear seasonally from your colony during the summer dearth, which happens in a lot of our nation, we see the same pattern where mites are actively moving on to 2-day-old workers. So they are highly attracted to these 2-day-old bees. This is what our early season data looks like in bar graph form. And you can see just the magnitude by which drones are harboring mites compared to their worker counterparts. And I always point out, it's actually fewer drones that are actually harboring. So there's fewer drones than workers in any of the studies, but they're harboring the majority of the parasite burden. And this is really typical of an aggregated distribution, which is what we see across parasites. Now, at the end of our study, we actually get this really cool data, which is we can see which bees were never observed with mites, ones that were parasitized during larval development, and the others that were parasitized during adulthood. Now, before Varroa destructor jumped from Apis cerana onto Apis mellifera, this is what every single honeybee colony, Apis mellifera colony, looked like. There's a bunch of non-parasitized bees interacting with other non-parasitized bees. They were rearing brood and taking care of the queen, trophallaxising with each other, and they were all non-parasitized. But the moment Varroa destructor enters our honeybee populations, this is what your colony looks like. Now, this is one of our colonies from an early season trial. I've omitted the drones simply to keep the schematic digestible. And out of 100 bees, this is what your colony would look like, representative of your colony. The majority of these bees weren't parasitized, and a minority of them were observed with a mite within 9 days. Now, my alcohol wash here is pretty high. It's 5.1%. Now, we know that's above the 3% economic threshold. But my absolute parasite burden, which is slightly different than your relative parasite burden, the alcohol wash, was 14%. What that means is, even though this pink bee right here might not have a mite on it on the 3rd day, it had a mite on it on the 2nd day. So it's still considered burdened by a parasite, and that's why it's the absolute parasite burden. And I really want you to pay attention to this number right here, because it's really important. And at this time of the year, again, drones are taking the majority of this parasite burden. They're harboring all of these bites that are occurring on the adult bee population. Well, we have a pretty severe dearth here in Maryland, and I went back into this very same colony and did the project again, a different trial on the same colony roughly 30 days later. And this is what your colony looks like once drones disappear. So our alcohol burden, our alcohol wash spikes in August. And this is typical in a lot of our country. It jumps all the way up to 14.6%. But our absolute parasite burden, the number of bees that at any one time are observed with a mite on them, skyrockets to almost 60%. So now your colony goes from 30 days prior, a minority of the bees are parasitized, drones disappear, and a majority of your workforce is now burdened by a mite. Remember, your worker bees rear brood, they trophallaxis with each other, both donating and receiving food. They take care of the queen. They take care of all colony tasks. And now it's impossible for them to interact with each other without having to interact with a parasitized bee. And now we've offered the opportunity for rampant viral circulation through multiple host-to-host routes and also through more vector transmission by mites. I really want to point out to everyone, these weren't dying colonies. This is what that colony that was parasitized by 60% looks like. And this is the really scary thing in these disease systems. Your infectious bees are often asymptomatic when you're looking at them. We pay attention to bees that are showing some sort of morbidity. Either they're quaking or they're falling over. They have crippled wings, something that's very ostensible that shouts out at us. But the fact is, any of these bees that it might moved onto, fed upon, and then left, you can't tell that it was bit by a mite. It just looks like a healthy bee until it's no longer healthy. So every single one of the colonies in our experiments crashed on us, but I really want to point out, this is 60% bit at this point. It's essentially walking dead. I just can't see it yet. And this is just another colony, same thing, that was heavily parasitized. It just doesn't look it. So conceptually, your colony looks like this over on the left early in the season, where most of the mites are on your drone brood and adult drones, and relatively few compared to the burden that the drones are hosting are on your workers. And it shifts late in the season. So what can we do with this new information? Well, we're teaming up with BIP, and it's still early in our season, but it's late for us to get a start on this. But we have been working for 2 years already, myself and my parents actually, and I'll explain that one in about a minute, with commercial beekeepers in Vermont to come up with a new sampling technique that might enable beekeepers to predict which of their colonies will become heavily infested come fall, all the way back early in the spring. And it comes down to by simply picking up a handful of these drones. What we've been doing is we've been pairing alcohol washes with hand sampling of up to 40 drones from the brood nest. And what we really wanted to know is, if I get multiple detections of mites on drones early in the season, is that predictive of which colonies will be heavily infested come August? Because in August, when I have a really high alcohol wash, I can't do too much with that information. But this time of the year, like right now in Maryland, I've already identified colonies that have lots of mites on their drones. I've already split and treated those colonies. And my goal is to go forward with these healthy requeened colonies before mites devastate the worker bee population. And we're going to team up with BIP to see how can we enable beekeepers to empower themselves with their beekeeping, both to remove queens that are not suppressing mite reproduction, and to keep viruses from circulating heavily in their colonies. But as of right now, I'm going to leave it to the group at Sentinel to do the correspondence with you. But if you are interested in information right now, my website's there. And if you click on the Citizen Science Project page, the methods that we're doing right now, a video that's online, and some groups that are actually trying this on their own as a citizen science project are engaged. You at least get a little extra information. And the reason this picture is here is we didn't have funding for this project. So I called my parents, and I know that sounds really dorky, but they got up once a month at 3:30 and drove from Eastern Connecticut up to Vermont and sampled bees with me for 3 to 4 days, and then went right back to work. And they've been doing this for 2 years. We're on a 3rd year now. And also pictured there is a girlfriend at the time and a beekeeper friend who would come up and give us a hand because, again, we didn't have funding, but we knew this was going to be valuable to beekeepers. That's the end of the presentation, and I will stop the share now. And I think I have a couple minutes for questions.
Awesome. Thank you, Zach. I'm sharing the link in the chat. All right. Does anyone have any questions for Zach?
I have a question. My question is, it almost sounds like you're coming up with an alternative way to test for mites on a relative basis by only using drones. Is there a formula for that? You said like 40, or is that another relationship we can have in testing for mites?
Correct. So we're in the process, which means we don't have any threshold numbers. We don't have universal best sampling practices yet, which is one reason why I'm here with BIP, because BIP is an unbelievable resource where we could rapidly expand this potential sampling methodology and to see, does it work across different geographic zones, which is the best number of drones to sample, and in which situations is it a really good sampling methodology. And I like the word that you used there, relative, because as of right now, the way I'm applying it in my own operation, I am using my detections relative to other colonies in the yard. So for example, if I hand pick up a bunch of drones from one colony and I don't have any mites, same thing in the next, same thing in the next, and then I get into one that has 2 or 3 drones with mites, well, relative to the rest of them, that one's heavily infested. And I take action on that one now. That's more field practical than scientific, but that's the infancy at which I'm in with the sampling method right now.
Gotcha. Thanks.
Yep.
This is Maureen. I have a question. So on our own, because this isn't part of our Sentinel sampling, we could do an alcohol or a Dawn dishwashing liquid wash on, say, pick up 40 drones from frames in our hives and throw them in the wash and see what shows up.
Maureen, I would absolutely love that. Now, just to be very clear, throw them in a different alcohol wash.
Yeah.
Yeah. Okay. Excellent. Yeah. And that is what the Citizen Science Project is. We have beekeepers. So actually some signed up from Canada, which was pretty cool. We have one master beekeeper for her project in Colorado that organized almost 30 beekeepers, which is amazing to do this. And we have a group in Virginia. And what they're doing is they're doing an alcohol wash and then they're hand sampling drones and they're submitting both data.
Okay.
And that's invaluable data because you all keep bees in different management styles and in different geographic areas. And one question that we have is, how universal is this, right? It may not be super universal, or it may be universal. And any of this data in preliminary work is extremely helpful.
And when they're doing that, they're doing the alcohol. Usually we do an alcohol wash on a half a cup of bees. Usually we aim for nurse bees, and do that, or we send them off to BIP or whatever. So it's not, I can't imagine this group is collecting 300 drones from each hive because to have the same number.
No. So no, and collecting 300 would be really a poor sampling methodology. So at most, you'd really expect 1,500 drones in your colony in a really large colony. So if you were to collect 300, you're now collecting 20% of your drone population. And by proportion, that's far more than 300 bees out of your 50,000+ bees. Okay. So we came up with an average of 40 is a great number to grab from 2 front brood frames in the center of the brood nest.
Okay.
And that method can be found on the on my website. There's a PDF that you can download. It's actually relatively quick. It's actually almost scary how adoptable this is, because, you know, we need to do alcohol washes to get early detections and to know what our mite levels are. But people are very quick and willing to sacrifice their drones to alcohol.
Yeah.
You know, they'll just, you know, quickly jump to that.
Great. Are there any more questions for either of the speakers?
I have a question for Zach. With regards to the mites on the drones, do you have any sort of feeling as to whether these mites are coming out as they emerge, or are they hopping on them afterwards? Yeah, you know, and is that why you want to collect them in the brood nest, because you're trying to get them upon emergence?
Amy, it's both. And the brood nest is for not because of emergence, but for a different reason. So mites are by a higher proportion on your newly emerged drones than workers. But in our studies, we're actually noticing that the mites are moving onto bees that were previously not parasitized. So essentially drones are gaining mites. What wasn't shown in the short presentation was we also did this work by sampling colonies at random from 2 commercial apiaries over the course of a season, so May through August in Vermont and New Hampshire. And we saw the same pattern, that when you pick drones from the center of the brood nest compared to hand picking up workers, you're going to get greater detections on those drones than you would on those workers. And the reason that we really want to sample from the center of the brood nest is not the likelihood of getting a newly emerged drone, but rather drones aggregate, older drones aggregate on the outsides of your colonies. So up in the honey supers and on those pollen frames on the outside, our data shows that those are the least likely drones to have mites on them. So our hypothesis is those would be poor drones to use in a sampling methodology. Whereas the younger drones that are going to migrate and walk around in the center of your brood nest, they're better candidates for a sampling methodology.
Well, that's very interesting, because I was thinking they might have been hopping onto the drones for dispersal, but that suggests otherwise.
Yeah, and that's a different topic, but we had drones in our studies within 3 days are in neighboring colonies. And by neighboring colonies, I mean on the other side of the yard, and they had to go around a shed, you know, as young as 3 days. And we're hand painting these things, so I know exactly which colony and when they were marked. And your drones are also the most likely bee to drift from one colony to another within an apiary. It's a different topic and a different study, and it's definitely for another grad student to work on. But that's another thing with our work.
Cool.
Awesome. Well, thank you so much. That was those were both great great talks. So for those of you who are interested in participating in either or both those projects, they are both optional. We will not share any of your personally identifiable information. And there was one more thing. And by submitting your samples, you agree to participate in the project, of course. All right, so if there are no more questions for either of them, I'd like to hand it off to Michaela to give a very brief reminder of how to get your app and dashboard account set up.
Okay, so I think we got it up now. And like I say, please don't hesitate to send in questions, and I'll answer those best as we can and get y'all set up. It might take multiple back and forths. Some of the questions that have come in, I might need more information or reminded about things. But if we go to the Be Informed website, after you see a reminder to take our law survey, which is still open if you have not taken it, we extended it to the 4th. You can go to Education Outreach, Sentinel Apiary Program.
And we need all participants for the last survey. So thank you.
Yeah,
Thank you.
and
Thank
if
you.
you could share that the last survey with your friends, that'd be even better. But on the right is these different one-pagers and different PDFs you can download that are going to help you with the program, including like inspection guides, but also protocols. And these first two relate to the mobile app and the web database. And so the first one, I saw a lot of familiar faces here, but there might be some new people that haven't participated yet. And this one, this is a one-page document that's going to show you the first things you need to do to get set up to use the database. After the first link there is to sign up and pay, and then it's directing you to research to the web to register for an account. So this is a step that we cannot do for you. We need you to do this because it's going to, get your link to your email. And so you go to the research.beinformed database system and click register. And this is going to be for both the mobile app and the web portal. And if you've already registered and you forgot your password from last time, click that forgot password button down there. Both of these methods, the forgot password or register, you will need to check your email for a message from us, from the server. If that email does not come through, it might get blocked for some reason. Let me know, and I can go in and see what's going on with that. And then once you register for an account, then the mobile app will work. The mobile app, you can download it without an account, but it's not going to work correctly until you have a login. To get to the mobile app, there's instructions here for iPhone and iPad or Android. And you can download the mobile app and log in with that same web email. And so that gets you like registered and access. I skipped one point. When you register for this account, there's a setup procedure that I need to do that you'll need to email me and let me know that you've registered. However, I am going through and looking at our registrations and seeing who has already registered and if they've paid, and then I go ahead and do that setup phase. So I did that Friday, went through, set up a bunch of people that had already registered, and we may still need to send an email out saying, hey, you're set up. But anybody that's registered as of Friday, you should be set up. And then once you're set up, this app quick start guide tells you a little bit about how to use the database. And Eric said briefly, so I'll keep it a little bit brief, but there's a few things I'm seeing in the questions that I think we can look at and that are covered in this document. So this is what the mobile app looks like when you have both logged in and you're set up to have access to the Sentinel database. There's a change here on this home screen. We have these records for operation owners, location, sample event, and management action. Last year, this row was called Beekeepers, location, sample event, management actions. So to use the app database well, you kind of need to understand the main concepts of it. And so this operation owner is going to be an entry that captures the entire operation. So if there's one apiary with 2 colonies in it, that's your entire operation. If you have 10 apiaries with colonies at each of those apiaries, but they're all managed in the same person or unit or business, then that's the operation owner. Some of the people in Sentinel, you're working with multiple beekeepers as a club, and so in those cases, those participants might have multiple operations that they're working with, and that would be the record here. And it's very important to keep everything in one operation under the same operation owner, because that's going to affect how the reports work and allow you to see the data in relationship to other colonies in the same operation. So I want to cover that. The location form, some people have reported that it works great, and then others are having a problem getting the pin to land in the right place, and it's highly dependent on the device you're using. So we're finding that the older tablets, the companies are not maintaining the tablets and phones as well as the newer ones. So if you have an older device and the app has any performance problem at all, you're probably going to see it in the map before other places. And so if the map doesn't work correctly, you can still name the location, let us know the GPS point. There's fields there where you can type those GPS points in. If you're able to get them some other way, we can put the GPS in for you. Just work with us to get those in there. And then the rest of this guide is going to walk you through what you might do to do these different steps of creating management actions, the colony numbers, all the helpers. This is explaining how you can make the colony number and it becomes reusable in the drop down. So the first time you type in a colony number, you can type it in correctly as it appears on the tag. You'll be better off in the future because you can reselect that and keep entering it without having to type it all out before. If you enter your colony numbers as 1, 2, 3, like this example, that's not going to be as useful to the lab as if you put the full number on there. And that will help them when they get the samples in at the lab to get their Varroa results entered into your database connected to you. So if you spend a little more time with these setup steps of using the app, you'll get more use out of it, fewer problems later on. And there's quite a few different things here to explain. This screen looks a little bit different this year for the brood diseases. In the past, we just asked you to record observed or not observed. Now you have the option to enter a degree of infested. So it's like it's really bad European foul brood, or it's just a small amount. And you're able to record that now on the app. You don't see that here. I guess I forgot to update that slide. And that's about it. I think that's all I'm going to go over right now. Oh, except for syncing. So when you use the mobile app, you get your data entered in there. It's only on your phone at that point, or your app. You got to click this sync with bit button for that data to go up to the server. And then you should be able to go to the web site here, log in, click on the user dashboard button, and then see your data there. And we'll be going over more over this as time goes on. If I need to work with anybody more directly to get you set up and make sure you understand, we can do that. And if there's any questions right now, I'll be happy to answer those or look at anything closer.
Michaela, this is Maureen. I just have a quick question. Do we record, there's a form for the provenance, the hive provenance, for those of us who are continuing. Are we doing that electronically, or are we doing that in paper?
What we would like for you to do is to use the management action. Now, let's see, let me go back and share my screen again. Well, in the mobile app, you can record management actions, and so the provenance form is mostly about what's the previous history of this colony.
Right.
And so if you could use the mobile app to record what happened to these colonies before the season started. So you could have a note about the treatment you did to the colony last fall, if it's an overwinter colony. That would be a good starting point. If you could record some historical note about the colony.
Okay, but I mean the actual form that they sent us on hive provenance.
Did that go
It-
out
th-
to you, Rachel? Or
Yeah.
have you, there is a
So-
form in there?
So
There's
we
not,
should do that on paper?
yeah, there's not an electronic.
Okay, that's what I was just
version of the
asking.
form, sorry.
And I noticed we got one copy of it, and if we're doing 8 colonies, so we just can photocopy it, I guess. And
I should have enough space for 8 entries on it, I think. If
there's
it
only
doesn't,
1, 2, 3, 4 colony tags. And then on the back, it says management actions. You can give the feed log, the treatment log, the honey harvest log, the equipment change log.
okay, sorry about that. Yes, if you can make a photocopy, that
Okay,
would be
we'll do. Not a
And
problem.
I think what we think is that if you put the management in the database, that you don't need to put it on the form. That would
Okay.
extra work for you.
Thank you.
Thank you.
awesome. Well, thank you, everyone. So we are now at time. So we are going to go ahead and stop the recording. Thank you so much for joining us for this May Sentinel Collective.